you're back. This is yet another lesson in my series of 7th grade science lessons, and this continues. In fact, this should be the conclusion of Chapter 3, Lesson 10.3. And today, we're going to take a look at mutation. But first, let's remember what we looked at yesterday, or whenever you last watched this video. For me, it was yesterday. We took a look at the 3 types of RNA. We saw that there is transfer RNA, messenger RNA, and ribosomal RNA. And all 3 of those RNAs are what it takes to build, super important, makes your muscles, makes your bones. Yeah, I can hear you saying protein. Good. This is how we make proteins. Okay, but that's not today's lesson. Today we move beyond our ribosome and our 3 types of RNA on to mutations. You have read that the sequence of nitrogen bases in DNA determines the sequence of nitrogen bases in messenger RNA, and that messenger RNA sequence determines the sequence of amino acids in a protein. That was yesterday's lesson. You might think these sequences are always the same, but they can change. A change in the nucleotide sequence of a gene is called a mutation. And there are, just like there were several types of RNA, there's several types of mutations. So if we take a close look, here's our original DNA sequence. So the 3 types, and if you're building a concept map, that's the way I would do this. I would say mutation, 3 types, and then include substitution, insertion, and deletion. Okay, shameless plug. I'm making a video on how to make a great concept map for Chapter 10. Since the concept maps are going to be your test, I'm not going to tell you everything, but I'm going to tell you a lot, so make sure you watch that. It'll be the next video after this one. Alright, so there's our original DNA sequence, and notice this one in the middle is C and G. Okay, here, now we have a T and an A, so it has been substituted. Kind of like when you're here and I'm not, but there's a substitute teacher. It's somebody different. But you aren't mutations, and the class isn't mutated. Well, sometimes it's a little mutated, but anyway, that is substitution. Then there's also insertion. This would be like if I was here teaching you and a second teacher was here teaching you. That would be pretty different, wouldn't it? And it might be better, it might be worse. That's the thing with mutations. Sometimes they're beneficial, sometimes they aren't. We think that mutations are the reason that we see changes in life forms over time. The reason that the ambulocetus, the walking whale, eventually turned into Basilosaurus, that great huge whale that was eating sharks. Don't you miss watching videos here? I do. So those things happened, we think, due to mutations. There may have been other reasons too, but mutations is one of the things we suspect. Usually they are not beneficial. Usually there's an enzyme that comes along and proofreads your DNA and says, oh, that's wrong, and it corrects it. Sometimes it doesn't, and the mutation persists. So here's an insertion where we have an extra nucleotide, extra base pairs have been added, and then finally, everybody knows about the delete key. So if you hit the delete key on your computer, whatever was there that you highlighted went away. Well, in this case, somehow, maybe a beam of radiation came through and just deleted those 2 base pairs. And so that would be deletion. And there you have it. It's that simple. Those are the 3 types of mutations that you can get with DNA: substitution, insertion, and deletion. Alright, the last thing that we need to cover in this chapter is kind of the coolest thing. It's called genetic engineering. It's one of the things that shows the greatest promise for our future, because we can actually go in and alter genetic code. In this case, I'm just going to step back out of the way and read this thing to you. You may know that chromosomes are made of DNA, you better know that, and are in the nucleus of the cell. sections of DNA and chromosomes that direct cell activities are called genes. Scientists are experimenting with genetic engineering, the biology and chemical methods that change the arrangement of DNA that make up a gene. That's your definition for genetic engineering. Genetic engineering is already in use to produce large volumes of medicine. Genes can also be inserted into cells to change how those cells perform their normal functions, as shown in the figure below. So there is a lot of promise in genetic engineering. There's many diseases that are genetic diseases. In class we talked about Down syndrome being caused by an extra chromosome. In theory, you could engineer a virus and actually make a virus do something good instead of something bad. It would go in, it would attach to every cell in your body, the 100 trillion of them, and it would edit and change and remove that extra chromosome and actually provide a cure. There's a lot of genetic diseases, and all of them could be cured by genetic engineering. It's not as simple as I've made it sound. There's a lot of steps to go through, and it takes a lot of skill. But one of the things we have done is this. Let's see, did I make that? I hope I made that bigger. Okay, so this is a bacteria cell. And what we did in a bacteria cell, we started up here, and you've got this DNA loop. And so you take the DNA loop out, you cut away a piece that you don't want. You put in this case, it's the human gene for making insulin. And if you don't know about diabetes, diabetes is a disease that means your pancreas isn't working right. Your pancreas doesn't create enough insulin, therefore your body cannot metabolize sugar. So you have to get extra insulin or you will die. So we have to be able to give people insulin. We used to give them the insulin made from slaughtered pigs. It wasn't really good, and it led to all kinds of other bad side effects. Now we can take this bacteria, we can plug in this human insulin gene. This is called recombinant DNA. Then you re-put that back into the bacterial cell, and now with the new bacteria cell, with this programming, it reproduces, and the thing that it does, it makes insulin. That's all it does. It makes tons and tons of good insulin that is designed perfectly for human beings. So there's your great example of genetic engineering and how it's really helped humanity and people that suffer with that disease. We can do artificial selection with plants. We can do genetic engineering with plants. We do genetic engineering with bacteria. And that gets us to this thing. This was the quiz I posted online, and some of you have already done it. I'm not going to do the whole thing for you, but I'm going to give you a little hint here. Remember that with DNA, A always has to go with T, and T always goes with A. C always goes with G, G always goes with C. So, what should go under the A? Come on, say it loud enough so I can hear it. Right, a T should go there. And what should go under the T? Yep, an A. You keep going just like that, and you too can read the genetic code. Oh, okay, I'm going to stop there. Thanks for watching, and stay tuned for the next lesson, which is how to make an awesome concept map. Thanks for watching.